All right. I guess you can hear me well. And welcome, everyone, to the very first week and very first lecture of our PNS Genomics course. Today, we'll mainly cover some basic introduction to the course, as well as some basic introduction to genome analysis. So with that, let's quickly get started. So I'll start by a self-introduction of myself. I'm John. I'm a senior researcher in the software research group and also a lecturer at ETH Zurich. My research interests mainly lie in the intersection of bioinformatics and computer architecture topics. You can get to know us, our group, our research using these websites. You can also contact me using my email address here. So you can access all these links on the website because these slides will be available also on the course website. So today we'll mainly cover the goal of this course, and also we'll cover what we expect from you, what we expect from students, so that you can have a successful run throughout the semester. And towards the end, we will also have a very brief introduction to genome analysis so that you can slowly get started. And also understand what type of data that we will be analyzing through this semester. So let's start with the key goals of this course. One key goal is for you to understand the computational steps and challenges in genome analysis, and then learn how to overcome these challenges with algorithm and hardware software code design. So this is essentially what we're aiming throughout this course so that essentially you'll acquire these when we're essentially going over the weeks, especially with your mentors. We have two PNS genomics courses offered every semester. One course is mainly about designing algorithms and also architecture design. And here, what we're aiming to accelerate the speed up the analysis of the corresponding computation of an algorithm or enable even new applications that were not possible to do so by algorithm and architecture design. The second course is also similar. There, we're also aiming to co-design hardware and software so that we can accelerate genome analysis, though this course can also include algorithm design to accelerate the genome analysis as well. Both of these courses are project-based courses. You'll be understanding and improving the cutting-edge techniques because we'll be showing you what sort of efforts, what sort of algorithms are currently being used, what sort of emerging hardware we have so that you can utilize the state-of-the-art mechanisms and even improve them. You'll have an hands-on research exploration with your mentors. So your mentors will be telling you what is expected throughout the weeks and what sort of research you'll be conducting with them. We will also have optional week lectures on genomics. We'll get to them a little bit later. And also, by the end of this course, hopefully you'll be improving your research and presentation skills. So yeah, as we said, this is essentially a hands-on project that you'll be mainly doing with your mentor. So what does this mean? Your mentors will be defining a particular project for you, and depending on the content of the project, it may be possible that you'll be designing a new algorithm and evaluating or simulating this algorithm and its behavior on a particular architecture so that you can observe the effect on the end result. So the end result could mean maybe there is a bigger application, a bigger package that we're targeting, and then you're tweaking, maybe revising a small part in this entire application, and then you'll be seeing how the entire application, the behavior of the entire application changes based on the changes that you're making. Um, the optional weekly lectures will mainly cover the basics to genome analysis. We'll be showing you what sort of data we're analyzing and what sort of challenges we have to analyze this data and what are the state-of-the-art techniques to tackle these challenges so that you are aware of maybe the entire genome analysis pipeline or the common steps in the entire genome analysis pipeline so that you can essentially better understand why we're doing the things that we're doing and why we're perhaps focusing on a particular problem that you may be working on. So these are optional. These are you don't have to essentially you're not required to follow these lectures, but it's always better to follow these lectures so that you learn the basics early on, so that maybe you make even better progress for your project. So what are the key benefits of taking this course? There are multiple key benefits. One benefit is mainly you'll improve your basic knowledge in genome analysis. This is important, especially if you're interested in getting into maybe biomedical research or doing some essentially biomedical work at industry. Then essentially you'll understand what is the role of genomics data in biomedical research and also in life sciences, and how we are treating this genomic data, how we're analyzing it, and how we are extracting insights from the genomics data. You will also gain some technical skills to analyze the genomic data and also in computer architecture, depending on the project that you're working on. Uh, Also, throughout the semester, you'll be discussing with your mentors critically. You'll be assessing whether you want to take a particular direction or follow a particular strategy as you are working on your project. So, as you're doing this, you'll be improving your critical thinking and analysis skills. You'll also have a familiarity with key research directions, especially in the genomics field. You'll be familiar with the state-of-the-art work, and especially if you are interested in continue working on this direction or working with us, then this means that you'll acquire maybe all the necessary basic knowledge before you start doing your own research in this field. And by the end of the semester, you'll be presenting your project to your peers, to your colleagues, and also to your mentors. And to do so, you'll be preparing some slides, and your mentor will be helping you to prepare those slides. So by doing so, you'll also improve your technical presentation skills on your project. Um, so then let's cover some prerequisites of this course. We usually get the question whether there is any basic knowledge in bioinformatics or genomics is needed. The answer is no, because we'll be giving you this basic knowledge um, as you basically process throughout the semester, throughout the weeks. You'll either be watching the lectures, so your mentor may ask you to watch certain lectures to gain the necessary knowledge for you to make a process, or during your meetings with your mentors, your mentor will be giving you the knowledge that you need so that again you can make a progress. So that's why you don't necessarily need a prior knowledge in bioinformatics, but of course uh, it can help you essentially make faster progress throughout the semester on your project if you already have a knowledge in bioinformatics, or if you follow the optional weekly lectures that we're going to be delivering this semester. But what we require is an interest in optimizing efficiency and solving complex problems. This is not just for bioinformatics, of course, this is for any maybe research problems. It is really required that you have an interest in what you're doing so that you can make a good progress. Uh, it is really good if you have a basic knowledge in C or C++ programming language, or maybe in Rust, or maybe in Python. So it depends on the project, but If you already have a basic knowledge in C or C++, this pretty much means that you can work on almost a wide spectrum of projects that we're going to be offering this semester. We really need you or require you to have a basic knowledge in bash scripting. This is because for most of you, you'll be accessing our HPC clusters, our servers, so that you can maybe download certain source code, compile it, run it in the server that is shared across many individuals. So then this means that you need to know how to use some job scheduling tools and use them fairly so that you do not block also anyone else. For all of these and also for you to access the server and so on to run the tools, you really need a good knowledge in bash scripting. So if you don't have it yet, so I'm not saying you should drop the course, but with a week of maybe a study on bash scripting, you can really get the most of the knowledge that will be needed throughout this semester. And this skill is not really necessary just for this course, but you'll see that the bash scripting is really necessary for many directions in computer science because you'll be accessing potentially the Linux-based servers, and then you're going to be running many different tools or compiling them. So the earlier that you get this skill, the better for you. If you have taken digital design and computer architecture or an equivalent course, maybe an advanced course on VLSI as well, it is really good because then this means that if you're interested in maybe some HDL implementation, some FPGA implementation, then this means we can directly offload you these projects, assign these projects to you, so that essentially you learn how we are designing hardware to accelerate genome analysis. With this, synergistic to the previous point, if you have an experience in either FPGA or GPU programming, this is really beneficial, but again, not essential. So then what are the course requirements and expectations from you? As I said, we have weekly lectures. These are optional. The schedule is on the course website. We will try to have weekly lectures. Those lectures will be premiered. Mainly, they are not going to be live streamed, but you're going to be able to watch them on YouTube. There will be a date that this lecture will be premiered. I guess this will be on Tuesdays, but it can change, of course. So if you just follow the course website and if you see a YouTube link that is recently uploaded, you can just essentially click to it and then see when it will be premiered. These are, again, optional, but strongly recommended so that you can make a fast progress on your projects. So then what are these projects, right? So we already asked you to fill a form on Moodle. You'll be telling us about your skills, your interests, and so on. And we're going to be looking at those skills and your interests, and we're going to be assigning you to your mentor or your mentor. So you can have more than one mentor as well. And then your mentor will get in touch with you, and then will tell you what sort of project that you're assigned on. This means you'll receive the further information in your projects from your mentors in the coming weeks. Ideally, basically, at the end of this week or early next week. But don't worry, you'll have enough time to make progress on your project. So then what is this time that you need to devote? So this is a 4 credits course, or 4 credit course. So this means by definition, you need to spend around 120 hours in this semester to get these credits from this course. And starting from this week, we'll have mainly around 10 weeks for you to work on the projects. So this means that you need to devote around 10 hours per week for this course. Then what counts as an hour, right? So you're going to be meeting with your mentor, hopefully regularly, maybe every week. This counts as time that you're spending for the course. So you're going to be working on your projects, right? So you're going to be implementing things, building things, designing things. These are all basically the time that you're spending for this course. But it's really essential that you make a progress and you meet with your mentors so that you make a progress to successfully pass this course. So to make all of these, to make a progress, it is really critical that you have a clear communication with your mentors. So your mentor will be telling you like your communication channel with them. Maybe you're going to use Discord or just use emails or Moodle or whatever. But it is really important that you ping your mentor very regularly, maybe every week or more than uh, once every week, right? So that you make progress, you update them, you ask questions so that you're not blocked. And your mentor essentially should be eager for you to make a progress. So they'll be hopefully responding regularly to your requests. But it's possible that essentially they are senior researchers or PhD students. So this means that they may have certain deadlines, maybe on some weeks that maybe they are going to be slow to respond. But regardless, just it is important that you ping them and remind them about your questions so that you make progress. So communication is extremely important. So then one requirement is to make a progress on your project so that you can pass the course. And the meaning of making a progress, it really depends on your communication between you and your mentor. But the second requirement is the presentation. So at the end of the semester, this is, I believe, is going to be in the week, actually, before the last week of the semester, I guess the week of May 19th, but we're going to be announcing the exact date later on. You're going to be presenting your project to your peers, right? So you'll prepare some slides and we'll pick a date that hopefully will it's good for everyone. So we're going to be asking you what sort of dates you're available on that particular week, and then we'll be allocating that time for the presentations. So this is basically the second requirement. You make a progress, but then you present, and all of these together is a complete package for you to pass this course. If you make a really significant progress and show good results in your project, and if you want to publish those results, we will support you for publication. And if you want to continue working on your project, you can do so by contacting your mentor, saying that you're interested in continue working with us so that you continue working on your project or any other project, even after the semester. So we're open to these discussions. Just contact your mentor or just me so that we can discuss the details about this. So as I said, we assign the projects based on your interest and skills. And your mentors will be contacting you to arrange regular meetings. And as I said, regular meetings are critical for you to make a progress. So these are currently the tentative weekly lectures. These will be available on YouTube. So these are the links to our course pages on the bottom. So if you just click them, you'll see the course schedule. So this is the tentative schedule for now. And here you'll see the YouTube links that will be premiered or live streamed on YouTube. So if you click them, you'll be accessing the YouTube and now watching the videos from there, including this one. We already offered this course in the previous semester, so these are the links. These are the links to the courses that we offered in the previous semester, as well as the YouTube playlist to the videos that we premiered or we live streamed last semester. So you can access them. You can already access them and watch some of the videos if you're not basically patient to watch them this semester. So you can access the previous semester's content and already start learning a lot. We've been offering these courses for many years, many semesters. So if you're interested in checking them, you can also follow these links. So then this is all about the course introduction. If you still have a question about this course, feel free to email me, send me a message on Moodle, whatever the way that you feel more comfortable, do so. And I'll try to answer them as quickly as possible. So now then let's switch gears a bit and then focus on the second part of this course, which is about some basic introduction to genomics. And let's start with covering what is a genome, right? To explain this, I need to show you maybe how our DNA is structured in our cells. So this is the famous double helix structure of DNA. And if you look closely to this DNA, you'll see these pairs of molecules or base pairs. We know that guanine G pairs with cytosine C and adenine pairs with thymine. And this DNA can be packed in a very compact form, making up the chromosome structure. The length of a single human chromosome can be from 50 million to 250 million base pairs as a single sequence of these DNA letters, as a single, basically, DNA piece, unfragmented way. And we have several of these chromosomes, and in total around 6.4 billion base pairs in a single human cell. And this is making up our entire human genome. So depending on the cell that we're focusing on, the number of chromosomes, how many copies we have from a chromosome can change depending on the genome we're looking at, right? So for mammalians, we have diploid chromosomes, meaning one chromosome from each of the parents. But for plant genomes, we may have more than one copy of each chromosome. This is known as polyploid. Or we may just have a single copy of each chromosome. This would be the haploid genomes. Depending on basically if this is bacteria, viral, or maybe mammalian or plant genomes and so on. It essentially the number of base pairs and number of chromosomes we have change. But for humans, we have trillions of these cells in our body. So we're basically storing a large volume of data in our bodies, if you think of in terms of the number of base pairs. And understanding the content and analyzing these genomes can reveal key insights for us that are useful for many applications. For example, it can enable personalized medicine, meaning we could prescribe or even design drugs by understanding how you will react to these drugs by looking at your genome. Or we can even now alter the content of your genome to solve fundamental challenges of life, such as aging. Or we could take a sample from anywhere, maybe, and then understand whether there's any harmful pathogen growing among us. And doing so, we could enable maybe rapid surveillance of disease outbreaks, like it very recently happened with COVID outbreak. And this genome analysis usually starts by taking a sample. This could be maybe a tissue or just any sample from any surface. If you believe that there are some living organisms on that surface, so you collect the sample, and then we have this process that is known as library preparation. So this usually includes purifying your sample. If you are doing DNA analysis, it just makes sure that the DNA is maybe copied many times. This is an optional process. And we insert special adapters at the end of these DNA so that they can talk to certain machines that will be generating the digital data from these biological molecules. So it includes basically many steps, and some of these steps in library preparation is optional, such as copying, such as amplification of each DNA molecule. But at the end of the day, what we have, so remember I told you single chromosome for a single human chromosome can be at least 50 million base pairs, right? So this means you have a single piece of DNA that is at least 50 million character long, 50 million DNA letter long. But due to some technological and biological limitations, we usually do not have these biological molecules as one piece, as a single chromosome piece. Rather, we will usually have them fragmented, a chopped version of them. And maybe we'll have many copies of these chopped DNA fragments. And these chopped DNA fragments are inputted to sequencing devices rather than a single piece of the entire chromosome. So then the sequencing devices take these chopped DNA fragments and generate some raw sequencing data. This could be, depending on how these sequencing devices are designed, they may be taking a picture of every character or maybe threading the DNA, the fragment into a tiny pore and then measuring some electrical signals as the threading happens. So depending on how the sequencing device is designed, it will generate some raw sequencing data, meaning it won't initially be directly some text-based ACTGs that is telling us the sequence of DNA that we have in your genome. But then we can convert this to text-based ACTG so that we can perform some analysis. And then we'll be covering what we mean by genome analysis in the coming weeks. But really, mainly these two steps, the last two steps, are the computational steps that we are mainly interested in. As I told you, we have many different sequencing technologies. In fact, there's even a very recent sequencing. It's not necessarily very recent, but now I guess it is getting some a bit more attention nowadays, which is SPX, or sequencing by extension technology. So basically, we have many different sequencing technologies, and then based on how they are designed, they all work differently. But at the end of the day, the goal of all of these devices is to tell you each character base by base, character by character in your DNA in a digital data form, so that we can look at them and generate insights from them. And in our group, we're looking at these newer sequencing technologies. So this is one such example. So this is a mini device that is a handheld device. You can fit it in the palm of your hand and carry it with you to do some in-the-field genome analysis. And of course, by looking at these newer devices, we believe that there is a bright future for intelligent genome analysis, meaning maybe we can carry these devices anywhere and then continuously extract insights whenever possible, maybe using our mobile devices. And to achieve this intelligent analysis, meaning fast, energy efficient, and accurate analysis, we'll continue asking the question, what sort of architecture, what sort of design, system design choice we should make. So we're asking whether an FPGA or GPUs are suitable, or maybe we just need a heterogeneous system on a chip to analyze the data that we're generating from the sequencing devices that generate our digital genomic data. Or maybe we're even asking what are the requirements of the computation. If we have a mobile device, maybe the requirement is we want to achieve a power efficient, energy efficient analysis with power efficient devices. And of course, to achieve the best possible result in terms of accuracy, energy, and speed, we need to look at the entire stack of computer architecture. This starts from the problem definition, even down to the devices level. And we're looking at this because we see a problem today when using general purpose machines for data analysis. So this is maybe a general purpose system, including maybe CPUs or even maybe GPUs, But then we're trying to use this device to analyze a data that may have some specific requirements for analysis. So since the general purpose machine is not necessarily aware of the specific requirements, specific needs of this special data, it may be slow to analyze this data. And there are, again, reasons to it, but one, again, main reason is uh, we can generate if you are using a special purpose machine for data generation, we can generate data at a very high throughput, extremely fast. But then, since we need to analyze a large volume of data with maybe specific requirements of analysis, this large volume of data may not be analyzed quickly on general purpose machines. So this creates essentially a bottleneck in the entire system in terms of time and energy. And this is not just about genomics, right? So the computing is mainly bottleneck by data today in many data-intensive workloads. So this is true for AI, ML, and graph processing, and also for genomics. So whenever we have a large volume of data that we need to move across the entire in the different levels of memory hierarchy in the system, meaning maybe DRAMs or in disk and so on, this means that you're spending a lot of energy and time. But then these are very important applications, and we need to gain insights and observations much more efficiently than ever before, because the rate that we can generate data is still increasing. And however, our capabilities to analyze this data is not necessarily increasing at the same rate. And this is because we see that data moment dominates the performance today in the system. So when we generate this data needs to move across many levels of memory hierarchy, meaning it will maybe be stored first in the storage, and then it will go to main memory, and then processor, and then back and forth, right? So we'll keep moving the data back and forth depending on the requirements of the application. And this is especially problematic because even a single memory request can consume more than 2 orders of magnitude energy compared to performing a simple operation in the system. So that's why, since we have large volume of data and since the cost of the memory request is large, we currently see that the data movement dominates performance today as a major system energy bottleneck. So this means we need intelligent algorithms and intelligent architectures that handle data well. And to achieve intelligent analysis, in the next lectures in the coming weeks, we'll be showing you how we generate the genomic data, how we analyze it, what are the alternative ways to analyze this data, what are the state-of-the-art ways to analyze this data, so that you'll be seeing this entire pipeline of genome analysis, and then you'll have an understanding of uh, what is a genomic data and how we can analyze it. So if you are interested in learning more, of course, we're making many materials available online. This could mean our published works or some videos that are available on YouTube. So this is one example here. We show the how we generate this genomic data and then how we extract insights from it, starting from the very first steps down to the very last steps. These are some recommended textbooks for you to have an understanding of the algorithm and data structure design in genomics. So another essential work from our group that shows you how we can accelerate the genome analysis, also by taking into the near memory designs into account. And there's another work where we review essentially the current ways, current steps in accelerating genome analysis by designing algorithm and architecture together. Also, how we can use processing in memory to solve certain steps, to perform certain steps in genomics. We also organize workshops time to time. This was a relatively recent workshop that we organized jointly with ReCamp 2023, 2 years ago. Some talks again for you to watch so that you can get an understanding of what we mean by genome analysis and how we accelerate it. Essentially, all these materials are already on the slides, and these slides will be available on the course page. So you can click those links and then go to the YouTube video and then watch them if you're interested. So with this, we covered all the materials that that are hopefully needed for this week. But if you still have questions, feel free to send me an email or send me your message on Moodle or any other place, so that we can discuss your questions. So with this, thanks everyone for watching this lecture, and hopefully see you soon in the coming weeks with other lectures.